Good day, this is the Professor, CBMD PhD, and today's Thursday, March 2nd, 2023. It is 4:06 and 16 seconds, a.m. Eastern Standard Time, and today's research, Pseudomonas aeruginosa. So let's hop to it here. As Pseudomonas aeruginosa, bacteria produce a molecule that paralyzes immune system cells. Date March 1st, 2023. Source: University of Freiburg. The researchers have discovered a strong immunological effect of the molecule lectin bacteria and a way to prevent it. As bacteria of the species Pseudomonas aeruginosa are antibiotic-resistant hospital germs that can enter blood, lungs, and other tissues through wounds and cause life-threatening infections. Many bacterial species use sugar-binding molecules called lectins to attach and invade host cells. Lectins can also influence the immune response to bacterial infections. Researchers have investigated the effect of lectin bacteria, Pseudomonas aeruginosa, on the immune system and found that isolated lectin bacteria can render immune cells ineffective. The cells are then no longer able to migrate through the body and trigger an immune response. The administration of the substance directed against lectin bacteria prevented this effect and led to the immune cells being able to move unhindered again. As soon as they perceive an infection, cells of the innate immune system migrate to a nearby lymph node where they activate T and B cells and trigger a targeted immune response. Lectin bacteria, according to the current study, prevents this migration. We assume that lectin bacteria not only acts on the immune cells themselves in this process, but also an unexpected effect on the cells lining the inside of the blood and lymph vessels. When lectin bacteria binds to these cells, it triggers extensive changes in them. Indeed, the researchers observed that important structural molecules were relocated to the interior of the cells and degraded. At the same time, the cell skeleton becomes more rigid. The cell layer thus becomes an impenetrable barrier for the immune cells. And so we ask the question, can this effect be prevented? To find out, the researchers tested a specific lectin bacteria inhibitor that resembles a sugar building blocks to which lectin bacteria otherwise binds. And through observation, the researchers discovered the inhibitor prevented the changes in the cells, and T-cell activation was possible again. And further studies are needed to determine how clinically relevant the infection of the immune system of lectin bacteria is to the spread of Pseudomonas aeruginosa infection, and whether the lectin bacteria inhibitor has potential for therapeutic application. The current results provide further evidence that lectins are a useful target for the development of new therapies, especially for antibiotic-resistant pathogens such as Pseudomonas aeruginosa. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.